Good morning. So I want to, in this pre-lab video, go over some of the inoculations and the one test that you will complete this week. So this week is, like many weeks, going to be very easy because all you're going to do this week, for the most part, is to inoculate several types of media that you will perform tests on the following week. So this is one of the things about microbiology, is that oftentimes we have to wait for the organisms to grow in order to see the results. So this week you're going to inoculate media that's going to allow us to determine whether a particular bacterium has the ability, for instance, to decarboxylate one of two different amino acids. So each of your lab groups is going to inoculate 3 different bacteria. I'm only going to do one for demonstration purposes. I've sterilized my loop, as is dictated by great sterile technique. Once that's cooled slightly, perhaps I even quench it on an uninoculated portion of this medium. I'm going to touch a colony. This is a bacterium called Enterobacter cloacae. So this is an enteric bacterium. We can either inoculate, this is a broth that contains lysine. The PDF will actually explain better all of the components of this medium. But I'm just going to inoculate this with that single colony. Flame again, close it up, sterilize my loop. In both the lysine decarboxylase, which is this medium, and the ornithine decarboxylase medium, the idea is to come up with an idea of whether any of these bacteria have the ability to decarboxylate those particular amino acids. This is an important diagnostic tool. Now, the tubes that you may be using that the TAs are making up for you for ornithine may not, in fact won't, look like this. So it's important that you label the media, because what's going to happen invariably is that some unfortunate lab group, not yours, but some unfortunate lab group is going to pull out 2 inoculated and fully grown tubes and not be able to tell which one is lysine and which one is ornithine. In this particular case, it's easy to tell, but it may not be in your case. So very important that you label these. Now, the other thing that's really important, in fact is critical to the decarboxylase reactions, is they need to occur under anaerobic conditions. One of the components in that medium is glucose. So in addition to having, I've mislabeled this one, this is actually ornithine. In addition to having the amino acid ornithine and the amino acid lysine, there's a small amount of glucose in here. What that does is it allows the bacterium to utilize that glucose and hopefully utilize all of the oxygen that's in there. Because bacteria that are capable of decarboxylating these amino acids can only do so under anaerobic conditions. And so the glucose is there just to allow an initial burst of bacterial growth to hopefully utilize all of the available oxygen. Now, to keep oxygen in this headspace from diffusing back in and preventing an anaerobic environment, we're going to place a vapor barrier on top of each one of these decarboxylase broths. So we have sterile transfer pipettes, and we have sterile mineral oil. So what I'm going to do, and you're going to do, I should crack that open before the video started. I'm going to take sterile mineral oil, pull some up into this sterile Pasteur pipette. You can tell I'm right-handed, I can't really do much with my left hand. Then, into each one of these already inoculated media, so that's another important point. You don't want to put the mineral oil on before you do the inoculation of the bacteria. And you also don't want to touch the glass portion of the tube. That way you can use this same transfer pipette to put mineral oil on all of your cultures. If I were to touch this to the lip, I would worry that perhaps I had contaminated. So what you want to do is put just, you'll notice that the mineral oil doesn't mix with the water, it stays on the top. You want to put maybe close to 1 centimeter of mineral oil. Oxygen doesn't diffuse through that mineral oil. So once the bacterium uses up the oxygen in the bottom portion of this, that's a very nice anaerobic environment. And if this bacterium is capable of decarboxylating the amino acid lysine, it can do so because the conditions, the anaerobic conditions, have been set up to do so. You want to make certain that you do the same thing for the ornithine. Again, I don't know if I flamed that last one, so I may not have used the best sterile technique. But I'm just going to add 10 or 15, maybe 20 drops. There's no set volume. You don't need a huge amount. You just need enough so that you have a vapor barrier over that. These are the only 2 types of media that we do this with. None of the other media that you will inoculate, certainly today or probably any other time in microbiology lab, will you use mineral oil on top of. So that's the only trick to inoculating that. You will also inoculate various other media with some of these gram-negative enterics like Enterobacter cloacae, such as the media that I'm going to inoculate next with just a single colony of Enterobacter cloacae that I've just touched. I'm going to go in. This is methyl red Voges Proskauer, or MRVP. Use your best sterile technique. Inoculate. Again, notice that I never set down the cover. That risks contamination. Put it back on, make sure that it's securely on. Sterilize the loop. Very simple. Same thing with the indole broth. I'm not going to talk too much about the MRVP test or the indole test because next week for the pre-lab videos I'm going to show you how to do these tests, how to complete these tests, because both the indole test, which I'm inoculating now, and the methyl red Voges Proskauer test require the addition of reagents after the bacteria have grown for 24 to 48 hours. So you have to inoculate these, label them very well so that you don't confuse media with similar colored caps. And then in the following week, you'll pull these back from the refrigerator. We'll put them in the refrigerator, and you'll add reagents to both the indole test to assay for the ability of bacteria to produce indole from the amino acid tryptophan, or in the case of MRVP, we'll do 2 different tests on that. And I'll show you that in the next video next week. The only other media that you need to inoculate this week is a very simple media. This is actually one of only 2 examples that we use in this laboratory of a completely chemically defined media. We know the precise makeup of this. The TAs for the laboratory have made this up to excruciating detail, so we know to the molarity every ion that's in here. And what we also very importantly know is that the only carbon source in here is citrate, citric acid. So we're assaying for the bacterium's ability to use citrate as a sole carbon source. And that's a slightly unusual phenotype among bacteria. So as a diagnostic test, this is a very important test. This is a slant media. You can, if you want to, stab this slant media, but it's not necessary. So I'm just going to take, again, I'm using as my example Enterobacter cloacae. I'm going to inoculate this agar media. And what I'm going to do is I'm using a loop, and I think a loop is the best way to go on this. And I'm just going to smear that across the top of this, all over the slant. I'm not going to bother to stab this, mostly because any bacterium, as it turns out, this is a trivial thing. Any bacterium that can use citrate is always only does so aerobically. But what's going to happen here is that as this grows, there's a pH indicator in here. You'll notice right now that it's green. If this bacterium can grow on this media using citrate, There's a pH change that will occur that will produce a beautiful royal blue color. It will change from green to blue. You may not actually see the growth on a Simmons citrate agar slant. Bacteria, even if they can use citrate, don't grow especially well on citrate, so you don't see much evidence of bacterial growth on here, but if they are growing on here, they will produce a pretty significant pH change, which causes this to turn blue. I didn't bother to label this one because there is nothing else that we can confuse a Simmons citrate agar slant with. So those will all be inoculated. They'll all be placed in the 37-degree incubator, and then myself, or more likely one of the TAs, will take them out 24 to 48 hours later and put them in the refrigerator. And when you come back in the following week, we'll record the results of the amino acid decarboxylation tests. Indol, we'll do the methyl red test, the Boakes-Proskauer test, and we'll record the results of the sodium citrate. The only test that you're going to do this week in lab where you'll actually see the results of is a test called the catalase test. The catalase test assays for an enzyme, appropriately enough named catalase. It's a very simple, pretty elegant, actually even kind of a fun test to do. It's really assaying whether a bacterium has the ability or has evolved the ability to actively deal with the toxic effects of oxygen. So even organisms that grow in the presence of oxygen, that require oxygen, that utilize oxygen, can find it toxic, because during respiration, some of the byproducts of oxygen respiration are not necessarily healthy. So things like singlet oxygen, superoxide radicals occur. And so some bacteria have evolved the ability to detoxify these very toxic reactive oxygen species. So some bacteria use an enzyme called superoxide dismutase, which will, as the name implies, dismutate superoxide. But one of the products of dismutation of superoxide is hydrogen peroxide. Hydrogen peroxide is not as toxic as a superoxide radical, but it's still very toxic. So bacteria, many bacteria that deal with the toxicity of oxygen have also acquired or evolved the ability to break down hydrogen peroxide. That's the enzyme catalase. Catalase breaks down hydrogen peroxide into water and oxygen. So this is a detoxification event. Now, we're going to use this test diagnostically in the laboratory. One of the chief ways that the catalase test will benefit us is that it helps to differentiate bacteria in the family Micrococcaceae, which includes the bacterium that I have right here, which is Staphylococcus aureus, from Streptococcaceae. This is Streptococcus mutans, the bacterium that causes dental caries. So I've taken a little bit of bacterial growth sterilely from this Staph aureus, and I now have a clean microscope slide. I didn't put any water on it, but I'm just going to smear that Staph aureus in a small little spot. I'm trying to get as much of that off of the loop as I can, so that you can see that. A little spot of Staph aureus. And then right beside that, I'm going to place a spot of Streptococcus. In this particular case, the dental pathogen, Strep mutans. And so I'm going to take a pull through there. This bacterium doesn't grow as well as Staph aureus. Staph aureus grows in the laboratory very luxuriantly. Streptococci don't grow quite as nicely. But I could easily get enough with a little pull through there that I was able to, can you see that? Make a little smear. Now, here's where the fun part comes in. I'm going to, you can't tell what can be seen here, what can't be. Remember that Streptococcus is on my right, and Staphylococcus is on the top. And I'm going to take, this is 3% hydrogen peroxide. This is hydrogen peroxide just like you would buy at the grocery store or the drugstore to disinfect wounds. I'm going to put a drop on top of the Streptococcus, just 1 or 2 drops. And I'm going to look, and I'm going to look. What I'm looking for is a reaction. And, you know, since I haven't told you yet what the reaction is, it's not obvious to you whether there's a reaction or not. So to compare that, I'm going to put it on the Staphylococcus aureus. These are live cells, or at least they are until I put on this hydrogen peroxide. Can you see that reaction? There is very vigorous bubbling. What's happening here is that the catalase enzyme, which is obviously present in Staphylococcus aureus but not Streptococcus mutans, is breaking down the hydrogen peroxide into water and oxygen. So the bubbles that are being given off are oxygen gas. So this is evidence of the fact that the Staphylococcus produces catalase, Streptococcus doesn't. Now, for your purposes, the reason why you want to remember this test is that when you get your unknowns, one of your unknowns is going to be gram-positive. And I can guarantee you that all of the gram-positives that are possibilities for your unknowns are either staphylococcal species or streptococcal species. And so once you've got your gram-positive separated out, one of the first things that you can do to differentiate is to do exactly this test, the catalase test. If you get bubbling like that from a fresh growth of bacteria, Staphylococcus. So it's going to be one of the staph species that we work with. No bubbles, it's one of the strep species. So you've kind of gone a long ways towards identifying your unknowns. So that's all for this week. Next week, we'll talk about how we're going to develop and finish off these tests. I will see you Wednesday, and then again in lab. Thanks.